right. Really light today in class. We'll see how many are watching. Yeah, I'll be glad when we don't have to do it. All right. We're getting people. Great people. Couple things. Lab next week, we're outside. Rain or shine. So looking at the 7-day forecast, it's looking pretty good, but weather forecasters they're about the only ones who can get things wrong and no one's mad at them. Who knows? I guess we won't even know until the day of. So it seems like weatherman says, yeah, it's going to be a rainy day tomorrow, or it's going to be a rainy day later today, and nothing. Sunny, hot. Or they'll say, it's a great day out there, and then we get 6 inches of snow and the city shuts down, right? Plan for it. We'll be outside. We're here on campus. We're just going to walk across over to the CHP or outside the CHP. We'll collect our data, you'll hand in your data sheet, and then you'll be free to leave. So usually on those days, we're done by about 3:30 or so. We will meet in the lab, we'll take the quiz, and then you'll get some instructions on what we're doing and how to do it, and then we'll walk on out. All right. So that's next week. Quiz. I think we have a quiz that's due tonight. Next week, we have an exam on Friday. And as I was looking through the exam material, trying to plan out when, what would be on it, where our stopping point is, and then also considering whatever stopping point I put, or I make, I want to be able to have at least a quiz on it so you can review the material before the exam. And I just don't think that's going to be possible because I'm not sure if we're going to get through all of this today, what we need to get through today. My suspicion is that we'll finish it on Monday, which means then the quiz would go live and you'd have until Wednesday to do it, and that doesn't give you a whole lot of time to look at the old quizzes. So what I did, or what I decided, was that we'll stop at populations. So when we finish the FST stuff and the Common Garden experiment, that's on our exam 2, and then starting with our populations, that'll be exam 3. So that means this quiz that's due tonight, that's going to be the last quiz that's on the exam. And then I also said that any of the old quizzes that you didn't take, you can still take them for late penalties, right? Or if you've got extra attempts left on those quizzes, you can take those up through Monday night or Monday at noon. So through the weekend, you can kind of take those, and then at noon on Monday, all the quizzes disappear, but the keys become available. That means you've got basically that week to study and prepare for exam number 2. All right. Those exam group 1 grades were actually pretty decent. So I'm hoping that some of you that might have been disappointed in your grades having this extra day or two to review quizzes and to study this material, hopefully it help your grade get those up. So I sent an announcement on Blackboard, so you should have seen it, you should have read it. Questions? Our exam is here in class, 8 a.m. All right, bring a calculator. Yep.
Did it start with the aquatic system?
It starts on right after it starts with water stress. So we did the thermal environment, and then it starts with water stress. So water stress in animals, water stress in plants, and then goes up through the intraspecific variation. And then our exam 3 will cover these populations. Let me see here, I can look at my presentations. We'll go through populations, population growth, and I believe life history strategies. So yeah, this next part is our population ecology stuff. All right. This is where we left off. We've got 3 fundamental descriptors: number of individuals, or density; spatial dispersion, so distribution across the landscape; and demographic characteristics, age structure, sex ratio, birth rates. All right. These describe our population. And we go back to this idea of ecological versus evolutionary population and the need to define boundaries, because our ecological populations, each ecological population could have a unique set of descriptors. All right. So what are these descriptors? How do we count? How do we estimate population size, dispersion, and these demographic characteristics? We're going to start with descriptor number 1, the number of individuals. So the number of individuals in our population is the population size. Population size is typically abbreviated with an N. N for our population size, and usually it's a capital N to represent population size. Now we do use lowercase N's and capital N's. We had that in the biodiversity lab, which incidentally is on this exam number 2. So it could take on some different meanings, but when we talk about populations, big N is our population size. If we start talking about population sizes through time, then what we do is add a subscript to it. So if we say N sub t, this is representing our population size at time t. And it's going to imply that our population size can change. Now, population size is useful. It's useful. It tells us how many individuals are there. But it doesn't necessarily tell us things about the interactions or how strong the interactions are going to be. For that, a better measure might be population density. Still a descriptor, it's still telling us number of individuals, but now our density is reporting number of individuals per unit area. Our 2 measures of density is accrued density and ecological density. The difference between these 2 terms is in how we count the area, or how we measure the area. All right? So for the accrued density, it's the total unit area. But for ecological density, it's only area that is suitable or appropriate habitat. So, let me diagram something here. Actually, instead of miles, we use kilometers. So say this, we set up a 10 square kilometer or 10 kilometer squared area. So total area in this area. $100 square kilometers. If we go out and measure individuals, we're going to do my stick figures because you don't really want to see me draw a 4-legged animal. All right, so we've got individuals living there. what we also have is a lake. All right. So our lake, let's say, represents 50 square kilometers. Can we, as humans, live in or on a lake? No, right? Unless you say, well, we live on a houseboat. Yeah, that's, we're going to say we're primitive humans. All right, so we can't live on that lake. So in this case, our crude density takes into account, or doesn't take into account, that lake area. It just takes into account our count for individuals, and it takes into account the entire 100 square kilometer area where we live. So you get what? 0.1 over 25, right? 1/25. Our ecological density only wants to include suitable habitat. So if the lake constituted half of our total area, our ecological density represents a much higher value. So, which one's more appropriate? Yeah, probably ecological density. Probably ecological density, especially if your sample size has a lot of inappropriate area or unsuitable area. Another equivalent is if we were counting, we will be counting ragweed, but if we did ragweed, on, let's say, the east side of campus, well, we have a bunch of inappropriate habitat. We have parking lots, we have sidewalks, we have buildings in the way, right? So we have to kind of subtract that to figure out what is our total area where those individuals can live. That's going to be our density, and that is our ecological density. In papers and stuff, do they distinguish between ecological and crude density? Yes and no. So oftentimes they do report the ecological densities without saying that it's ecological density, but in some place in that paper or in the methods, they'll say, we counted up all the areas where they could live, and that represents our total area. So up here, I also have another example, a ranch. Same idea. You've got 10 acres, you've got a 5-acre lake. If you are working towards deer management, you can see that our densities, crude versus ecological, represents a very different kind of view. In the crude density, it looks like we actually have less deer per area. But if we account for that lake living there, now we have 1 deer per acre. All right? So density is often a more accurate representation of population size because it tells us more about potential interactions. All right, if you have 100 deer in those 10 acres, those 100 deer are competing for the same type of food or the same limited food with everyone else that's there. But if we change it instead of 10 acres to 100 acres, now 100 deer, very different picture. Very different picture. So number of individuals, population size. So how do we do it? How do we calculate it? 2 different ways. Just kind of depends on, there are a couple different ways. It depends on what type of organisms we're trying to count. So the first way is just to do a total census. And what this means is we're going out and just counting individuals. All right, we're counting individuals, and maybe with one of those clickers, counting them every time we see it, and at the end of the day, we look at our total counts. This is really good if we have discrete individuals. where you can easily define an individual. Yep, that's a tree. That's one tree. Click. That's a second tree. Click. Pretty easy. If they're conspicuous, so they're easy to see. They're not hidden. They don't have camouflage and so forth. They don't hide during the day. And organisms that have limited mobility. So if they have limited mobility, that means we can kind of go out and when we count them, we can mark them in some way so that we know not to count them again. Or if we're running, let's say, a transect, we don't have to mark them because we're not going to ever walk back through the same area. We're just going to start counting every single individual. They don't move. So once you pass them, you have no worries that they're going to jump up and move up to the front of the line where you count them a second time. All right, so total census works. But it only works for certain types of organisms with a certain type of characteristics. So what are the bad characteristics? Well, if we have non-discrete growth, so vegetative growth, it makes it hard to really identify an individual. Because oftentimes in these growths, they kind of spread out and maybe put down roots. I'm going to talk about plants. Maybe they put down roots in different parts of the soil. So do we count where each one goes into the soil as an individual? Or since they're all connected, that's an individual? Who knows? Other plants, their roots are all connected and then they send up different shoots. So you might think you've got different individuals because they have different shoots, when in fact all their roots are connected. And we haven't even talked about colonial organisms like coral. Right? Where does one start and the other begin? Or where does one end and the other begin? So non-discrete growth, kind of hard to do a census, total census. Also, if our organisms are hard to find or highly mobile, it's really hard to count them. And it's really hard to make sure that once you count them, that you don't recount them, or you don't count them a second or third or fourth time. So total census works with some, but not all organisms. The second method is to use mark recapture experiments, and this is what we did in lab. These are good for discrete and mobile organisms. So organisms that are easily identified as an individual, and these are going to be mobile. All right, so we're going to go out, we're going to count individuals, and as we count, we're going to mark them. That way we don't double count them. And then we're going to let them mix back in populations and take a random sample. And the proportion of marked to our total in that random sample reflects the proportion of marked in the total population size. All right, so we went over the equation. We can use the Lincoln index is what we've used, and that's what we'll be tested on. All right, so you've got your marked over the total population size. That's the ratio that we're trying to approximate. And we're going to do so by marking individuals, M, releasing them in the population. And then when you recapture them, we've got total captures, and we have those that had been marked previously. Those are our recaptures. So this ratio should reflect the ratio of marked in our total populations. We can rearrange, so cross-multiply, to get our population size on one side of the equation. And remember, we estimate population size at the time of initial capture. That's what we estimate. All right, so know these equations, know how to use them. We'll have questions where it's population size, or we go out and mark 500 individuals. We recaptured 1,000 of those recaptures, 200 are marked. What's our population size? Know those. Know the assumptions. They're in the lab. So you should know those assumptions. So this is kind of that mark recapture lab spans 2 different exams. Our exam 2 is going to test you on this stuff. All right? The exam 2 is going to test you on this stuff. Exam 3 is more about when would we use mark recapture over total census. All right? And then final exam will probably have a question that has you estimate population size. It's a useful skill for your major field tests and for any, there, if you take the GRE subject test, it's on there. It's on. Lincoln index isn't the only method of estimating our population size for mark recapture experiments. So FYI, remember we did the Schnabel method. That's good when we do repeated recaptures. But then there are also some other methods, a little bit more complex methods, that not only allow us to estimate population sizes, but can allow us to estimate survival probabilities and capture probabilities and so forth. So, rather interesting field, this mark recapture stuff. Third method of estimating population size is with an index of relative abundance. Now notice that I didn't say measuring n, I said estimating. Because the index of relative abundance isn't going to be counting individuals. It's not going to be doing mark recapture and looking at proportions. Instead, what we're going to do is utilize evidence of individuals' presence. And then using that evidence, we can come up with a relative size. And the relative size isn't, oh, well, we've got about 100 or we have 200 here. It's relative to something else, to some other time point or to some other habitat. So we don't ever really have any number. So for example, we go out, we're interested in identifying population sizes of, let's say, raccoon. All right? And we're doing this during the day. So what we do is we have two different habitats that we're sampling. We're sampling during the day and we go around and we look for latrines, areas where the raccoons will defecate. All right? And then we count up the number of latrines, or we count up the number of scat that we see, the amount of scat. Habitat with more scat likely has more individuals. So we would say that habitat has a higher population size than habitat number 2. We didn't come up with a number, we just assigned a larger size or a smaller size based on the evidence. All right, so the index of relative abundance, I've defined it, is a quantitative measure of the relative size of a population using indirect evidence of the presence, absence of individuals. So our indirect evidence, again, scat, you can talk about rubs for deer, you can talk about scratch marks, all right? For birds, you can talk about calls, just going through and recording every time you hear a call, all right? You don't have to see the individual, you listen to them. That's our indirect evidence. When is it useful? Anytime individuals are difficult to count, we can utilize this, or maybe the total number doesn't really matter. So if we are undertaking management decisions for an area, say wildlife management, and we're trying to manage a deer herd, we might not need to know actual number of deer. We can go out and we can estimate relative size every year to see, is our herd increasing, is it decreasing, and so forth. If it's increasing, hey, maybe that's a good thing. Maybe that's a bad thing. Depends on what your goal is for that management. But again, our end didn't really matter. We're just trying to increase or decrease populations, in which case our index of relative abundance will work. Questions? All right. We're going to be talking about descriptor number 2, but we'll take our quiz, attendance quiz. All right. So the quiz has 2 answers. Two answers here are hearts and spades. The 4 suits. Hearts and spades. Hearts and spades. All right. All right, ready? Hearts and spades. Other. All right, ready? All right, descriptor number 2, spatial dispersion. Dispersion is the pattern of spatial distribution of species in a habitat. Dispersion, that is our spatial distribution. Dispersion is different from dispersal. Our dispersal is movement away from a natal habitat. Dispersion is the pattern, the spatial distribution. All right, so don't confuse those. There's 3 patterns that we can see, and these are not patterns that are discrete. All right, there's a continuum, and we could be, really on one side, or we could be at the complete opposite end of this continuum. So the 3 patterns that we see is a uniform or regular pattern. We could have a random distribution, or we could be clumped or aggregated. All right. So uniform or regular, we could be random, or we could be clumped or aggregated. So in these figures, assume each point represents an individual. And then if we kind of take a snapshot overfed view and we plot out the individuals in this habitat, it could be clumped, where all individuals seem to be close together, and dotted throughout our landscape. They could be uniform or regular, where it's like they're evenly spaced, equally spaced. Or it could be random, completely random. The amount of dispersion is quantified with a variable called the coefficient of dispersion. It's abbreviated CD. Coefficient of dispersion, CD. The coefficient of dispersion is essentially the variance to mean ratio. So we can calculate, let's say, distances between individuals. Right, and then we have an average distance, and then we have the variance of that distance, where that variance tells us how much variation we have in those. If our variance and our means are equal, that means the variance is equal to our mean. When we do that ratio, we get a coefficient of dispersion equals to 1. That suggests a random pattern, random distribution pattern. And the reason is that a random pattern follows a statistical distribution called a Poisson distribution, which has the property that the variance and the mean are the same value, or they're equal. If our coefficient of dispersion is less than 1, then we have a uniform or regular pattern. If our coefficient of dispersion is greater than 1, then we have a clumped or aggregated pattern. All right, so these are the terms you should know. This is the type of pattern that you should know. We're going to go out in lab next week. We're going to collect data on ragweed, and we're going to determine what the dispersion pattern is. But before, we can't just look at the coefficient of dispersion. What we have to do is first test, is our value equal to 1? If it is, it doesn't matter what that value is. It could be 0.97, it could be 1.3 or 1.5. Statistically, we just said it's basically 1, so we have a random pattern. But with our statistical test, if we can say, yes, this is not equal to 1, then we look at our coefficient of dispersion and decide, are we clumped or aggregated, or does it suggest we're uniform or regular? So we'll do that in lab. In lecture, what we're going to do is talk about how we can come up with these 3 different patterns. So we're going to start with the clumped or aggregated dispersion pattern. Clumped or aggregated occurs, or will give us a coefficient of dispersion value greater than 1. This means that the average distance between our individuals is small relative to the variance. So you have a bunch of values, a bunch of distances that are really close to, that are really small because individuals are close together. But then you also have distances that are very large because these individuals have a long distance from these individuals that are over here. So you've got a range, a much bigger range of really close individuals, really far individuals, leading to a high variance. In layman's terms, if we have a population that is exhibiting this pattern, if we find an individual, then we have a high probability of finding a second individual nearby. So finding one individual makes it more likely that we'll find another individual. Now, what types of factors favor this pattern? And again, you can have all of these, you can have one of these, maybe each of them is very weak, but it leads to this type of pattern. So it's not the factors cause the pattern, but the factors that favor the formation of this pattern. So number 1 is transmission and dispersal stages. So for transmission, we're talking about parasites and infectious agents. So bacterial and viral agents. How would this transmission and dispersal stages kind of produce this pattern? Let's think about it. How would we get this pattern based on the dispersal stages? We'll start with dispersal stages. I can call on people.
Like you're more, like you get sick and have a higher inoculation bacteria, like grouped.
I can see where you're going,
Yeah.
yes. So transmission's easier in a group because if one individual goes there, they're putting out viral particles, all right? It might not infect everyone, but the more individuals that are there, you increase the likelihood that they inhale those particles. Yes. All right, so yeah, for transmission stages, yeah, same thing with parasites. Parasites rely on transmission stages being eaten or consumed. All right, and they may be in one area themselves. Individuals that swim through that area are going to be more likely to get infected. So yeah, that's for our transmission spot, and we see that. I mean, this is just a fact. What about dispersal stages? So again, we said dispersal is movement away from the natal area. How would dispersal stages affect this pattern? Name a dispersal stage. Any dispersal stage. Name one. What's that? Okay. Yeah, let's avoid that. Let's avoid pollen. Let's avoid pollen. What's related to pollen? Well, not really related, but an end product of pollination. Okay, keep going. What does that produce? Seeds. 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 Dispersal stage. Seeds. In plants. How would plants produce this pattern? Yep, if you drop acorns, do acorns get up and walk away? Do they fly away? No, you're going to have a huge number of seeds underneath that tree. More likely to get younger trees growing up close to the parent. All right, so yeah, we do see quite a few plants that exhibit this pattern because of their dispersal stages. They don't get far away from that parent plant. Factor number 2 is habitat heterogeneity. So what is habitat heterogeneity? What do I mean by that? What do I mean by that? What do I mean by that? Logan. Well, we're talking about individuals. Habitat heterogeneity is more in reference to the physical factors and physical resources. So what do I mean by habitat heterogeneity? It's on exam 2. Anyone want to volunteer? Or should I just assign this for homework and then have a quiz? Okay, so you're saying because the habitat covers a wide area and we have a lot of variation in that area in terms of the distribution of our resources and factors. That's our habitat heterogeneity. So we've got areas that are good habitat, areas that are bad habitat. All right? And our organisms tend to live in those good habitat areas. They're clumped, they're aggregated. What about social patterns and reproductive behavior? Does that have an effect? Yeah, it does. So gregariousness is, yep. So gregariousness is like sociality. Sociality. Do the organisms socialize and form groups, form herds? All right, so yeah, some organisms, they have these types of social patterns, and thus when we look at distribution of individuals across the landscape, you're finding those herds, those patterns. All right, and then reproductive behavior, this might not be year-round, so you may exhibit one type of pattern that maybe is random or uniform during parts of the season, and then during mating season, all of a sudden, you start getting this type of pattern because you have males all flocking to those limited females that are out there. And then weather and environmental conditions can produce this pattern. This is related to our law of tolerance. And we talked about what happens when we fall inside our stress zones or into the zones of intolerance. What happens to our individuals? Well, one way is that they can, if they don't die, one way that they can get around that is to move. So if we have extreme weather, if we have environmental conditions that produce certain areas that are now inhospitable, all right, our organisms can move and congregate together, or they can congregate in those areas that provide shelter. This part, this last one, weather, environmental conditions, much more related to the abiotic factors, abiotic conditions. So they're tied to our law of tolerance curves that we talked about. All right, clumped or aggregated. Random. So clumped or aggregated is the most common pattern in nature. The other two patterns, random and uniform, are much less common. All right, so random pattern is when our coefficient of dispersion is equal to 1. Individuals are randomly placed on a landscape, so if we find an individual, it does not change our probability of finding another. It doesn't go up, it doesn't go down. Unlike our aggregated distribution pattern, the random pattern has some requirements. So the first requirement is that we have a uniform environment. We have a uniform environment. So in our area, the individuals see the environment as being the same. We don't have habitat heterogeneity at this point. It's entirely uniform. Our individual can live any place within that habitat area. And this also applies to the temporal scale. So we're not going to see the short-term environmental conditions or weather conditions change that would cause them to move and aggregate together. But again, remember, I talked about saying that clumped or aggregated can happen during certain types of years, and then random patterns can happen later on, or uniform patterns can happen later on. That could change. Our pattern could change. But if we see a random pattern, we've got a uniform environment happening right now. And we have neutral intraspecific interactions. Intraspecific is within a species. All right, so interactions between members of the same species. They're neutral. If we didn't have a neutral interaction, then we have one of two types. We either have a positive interaction or a negative interaction. If we have a positive interaction, then the presence of one is going to favor the presence of another. They're having a positive effect on each other, which makes it more likely that both of them are going to be there. If you have a negative interaction, then one individual is trying to exclude the other. So if we find the one, we're less likely to find that second. In order to get this random pattern, we have to have a neutral interaction, or neutral intraspecific interaction, because one individual can't affect the probability of finding another. That's part of our definition. All right. Our third pattern, last one, uniform or regular. This is when our coefficient of dispersion is less than 1. In this pattern, our distances between individuals are approximately equal. We do have some variation, but they're approximately equal. We do have factors that favor this pattern. Strong intraspecific interactions. And these will be negative intraspecific interactions and start to favor this type of pattern. Why? Because the presence of one is going to repel or repulse other individuals. Right? So the presence of one is actively excluding another. And it's going to have to be a strong intraspecific interaction to try to form this type of pattern. All right? The other factor is dispersal stages of some plants. And this almost seems contrary to what we talked about with dispersal stages helping to form this clumped or aggregated pattern. But again, this isn't all plants. It's going to be certain plants that have certain characteristics, certain qualities that can generate this type of uniform dispersion pattern. And we're going to talk about that, but I think what we're going to do is talk about that on Monday. So on Monday, we'll have a quick review of these patterns and then move to this Jansen-Connell hypothesis that explains why some plants have a uniform or perhaps a random dispersion pattern. All right? Any questions? Nope. All right. Oh, here it is. Open image in new tab. This is small. You've all seen this. This is a type of uniform pattern for these emperor penguins. They're all about a wing's length apart because they are extremely territorial at this time. So that when you do overhead surveys, they look something like this. That is a very strong intraspecific interaction. It's a negative intraspecific interaction. All right. Have a good one.